First of all, I thank the organizers for inviting us. I say us, but it was actually Mike Levin who was actually invited, and he just sent me to fill me in for. So I'm sure he would have given a much more exciting, high-level talk spanning a broad range of ideas, but you have to bear with me for my sort of a low-level, more focused technical talk. Anyway, speaking of multiple scales, today I'd like to talk about how we try to understand collective behavior and intelligence in developmental biology. Now, I'm not a biologist myself. I come from the modeling side. I was trained in complex systems. So I'll try to illustrate some of the ideas that we have been using very concrete, minimal models. But to set things off, I would like to show this video. What is it showing? I'd like to show this video of collective behavior that occurs during embryogenesis. It's a bit different from what we have seen during the last few days, but it's just as beautiful as all these examples that you have seen, if not even more beautiful. Of course, I'm biased. But let me just play this for you. It will just run for 2 minutes. It's what you're looking at is a embryogenesis of frog. So it will just start from one cell and it will just develop into a full-scale tadpole.
The fertilized ovum undergoes rapid divisions and forms a blastula. Each division normally takes about 30 minutes. The blastula develops into a gastrula. Neural folds develop and fuse to form the spinal cord and brain.
Why did they have the music?
I know. It's so dramatized.
Eventually, the embryos hatch as free-swimming tadpoles, or larvae. The tadpoles undergo metamorphosis, and tiny frogs emerge that eventually grow into adults.
I guess the music is to say that we still don't fully understand this process. So anyway, as you can see, it's a very different beast that we are looking at, right? And it's a bit different because the size of the collective itself keeps changing over time, and the shape also keeps changing over time. So it's as if the target at the various stages of collective behavior itself keeps changing. And also what you saw was just the morphogenetic collective. And there are so many hidden scales. There is this genetic scale, there is this biochemical, this biomechanical, and even bioelectric, which I'll be talking about today. So the grand challenge for development, one of the grand challenges for developmental biology is how do we understand this disparate collective phenomena in developing systems, right? Using a common language, right? So that would make it a bit more appealing to people like us here, because we want to understand collective behavior through a single common lens. So, and one of the motivating ideas that we used to try to answer this question is this paper that Mike wrote just last year, is this idea that life may be just trying to solve the same kind of problem at different scales, whether it is the transcriptional scale, metabolic scale, biophysical scale. It's the problem of navigation in a dynamical surface. Like we want to get from one point to another point, which is the lowest point of the free energy, which is the safest point for life to dwell in. So the same kind of navigation problem that's occurring in all of these scales. And Mike makes this bold hypothesis that evolution may be searching for these high-level strategies rather than the low-level hardware that we find in life. So we kind of use this idea as an inspiration for characterizing or thinking about collective behavior. So I will get to that. But I also want to talk a little bit about the kind of modeling approach that we have been using. At least I have been using in the lab, because there are different modeling people in our group and different people use different modeling strategies. So if you think about models as falling on a spectrum, on one end we have these phenomenological models, like the spin models or V-shaped models that we saw before. And on the other end, we have this highly detailed biophysical models that takes into account each and every little factor, the genetic factors, transcription factors, and everything. Our approach that we have been using tries to combine some of the genetic features of the phenomenological approach and also incorporate some biological constraints that we find in biological systems. So it's a kind of a hybrid modeling approach. So I'll be walking you through 3 different examples of this approach. And they are all minimal 2-scale models of collective patterning and decision making. So in the first example, we have a simple one-dimensional model of axial pattern formation, which has a physiological scale and also a genetic scale. And the second example, we have this 2-dimensional model that tries to solve bioelectric control of morphogenesis in a certain model system that we'll talk about. And in the third example, we use this model that involves both the bioelectric network and an electric field that develops a vertebrate phase pre-pattern in frog embryos. And the idea that I would like to drive home is that we can think of collective intelligence as these high-level causal influence patterns by which the units of the collective collect across scales. So it's the patterns by which this unit integrate and segregate over space and time. So that's the common language that I'm talking about. So all of these 3 models attack very different problems in development biology, but by using this high-level causal influence network strategy characterization, we can characterize collective intelligence or behavior in these 3 systems. We can look at all this through a common lens. So that's the kind of broad idea that I'll be trying to drive home. So let me dive into the first example. The first example is axial polarity development. So as you saw in the video, as the frog embryo develops, at the end of the gastrulation stage, you saw a sort of a bump at one end. So that's the axial pattern. So that bump develops on one pole of the embryo, but not on the other pole. So that's the stage where the embryo knows that there is an anterior pole and a posterior pole. So we tried to build a very simple model that demonstrates this qualitative phenomenon. So the idea is that how does an embryo develop from something very featureless to something that has this axial pattern? So to do that, we designed a very simple single-dimensional minimal model that has a physiological scale and a genetic scale. So it's a single-dimensional chain of cells, and each cell has a physiological state that might represent, say, the concentration of a morphogen, or it could represent anything you want. And each cell has a genetic network, and it's the same genetic network in all the cells. And the biological constraint that we incorporated is that the cells only talk to the neighboring cells. So that's a non-neural tissue. That's what embryos are. And they are constrained by this local connectivity. So this is the model in more detail, but I think for the purpose of this talk, this level of detail is not necessary. So I'm even skipping some of the equations, but if you're interested, I can go over it. But just a little bit more detail again. So each cell has two states, a physiological state, and it also has a cell type state. Both of these are scalars. And each cell has a genetic network. And we also incorporated planar cell polarity assumptions. So there is a sense of direction to this gene network. And this part of the gene network influences this gap junction. Again, the cells communicate through gap junctions. These edges represent gap junctions, and their permeability can change over time, so it's dynamic. And the GRN controls the permeability of these gap junctions differentially using a planar cell polarity sort of model. And in fact, this model also has more factors which I'm not showing. That's not necessary for this talk, but this is the kind of model that we are using. So what we did was we used gradient descent-based optimization to optimize this model so that when you start this model with uniform initial conditions, so all GRN states are 0 and all the physiological states of all the cells are 0. If that was the case, we wanted the model to develop a simple gradient pattern. It should start from here and should develop a simple ramp-like gradient. So that's your axial polarity pattern. So we were able to do this. We just used PyTorch to do that. And this is what it looks like. So the horizontal axis is the cell, so it's just a simple 12-cell model. And the vertical axis is the normalized cell activity level. So this was the initial state. It's just horizontal. And the red line is the target that we wanted it to converge to. And you can see that the final state looks like this. So here's a video. I'll just quickly show this. So it starts from a uniform state, and then what happens is that from a uniform state, it develops this state. And here, the thing is, we start the network with completely homogeneous state. There is no dynamic symmetry breaking that you might find in Turing patterns. There is structural symmetry breaking here because the cells at the end of the chain have only inputs coming from one side. So that's the part that breaks the symmetry, and that's why you see initial activity at the ends of the chain. And so if you just run this, you see this sort of stripe-like thing in the intermediate state, and then it slowly changes, and towards the end, it coalesces into this gradient pattern. So this is just to show that we now have a model that develops this simple axial polarity gradient that starts that it develops from homogeneous initial conditions. Now, the question that I was interested in, that we were interested in, is as simple as this model is, how does it work? Because it has so many different factors. It's a one-dimensional model, but it has two scales. It has a physiological scale and a gene regulatory network scale. And that GRN had about 9 genes connected in very complicated ways. So how does this complicated network develop a simple gradient, such as the axial polarity pattern? So to do that, we considered a simple but very expensive to compute measure of causal influence that's represented by this derivative over here. This derivative keeps track of changes in the physiological state of some cell due to changes induced in the genetic state of some cell over space and time. I say over space because the source and the target cells could be very different. It could be arbitrary. And I also say it's over time because we can keep track of these changes over multiple different timescales. So when we compute this, what happens is, of course, the network is fully connected in the sense that the signals from one cell can reach every other cell if you run it long enough. So if you just draw this network, you will just see a fully connected network. But to keep things simple, what we did was we took the strongest causal influences, and then we visualized it as a network, and we saw how that network changed over time. And that revealed some interesting patterns. So I'm showing this network at 3 different time scales. So at the very smallest time scale, as you can imagine, the causal influences are limited to neighboring cells, right? At very short time scales, signals only propagate from between neighboring cells. And it just happens that these causal influences are negative for whatever reason. But if you run it over longer timescales, you see some other interesting patterns emerge. Now you see positive influences shown by these blue arrows also emerge. And at the longest timescale, so this is the timescale of the entire simulation period, what you see is that all the positive influences cluster in the anterior half of the cell, and all the negative influences cluster in the posterior end of the cell. And more, the cells in the posterior end are completely controlled by the cells in the anterior half. So this network, this complicated network basically boils down to this coarse-grained network, where you see this super cell representing the anterior half that positively influences itself, and it negatively influences the anterior half of the network. And this we can understand, right? This is interpretable because it kind of helps explain why, from an homogeneous initial state, we get a gradient that is positive in the anterior half and negative in the posterior half. So this macroscale pattern is what is one example of characterizing collective behavior of a complicated system like this. And so this is the first example. I'm going to move on to the second example, but do you have any questions? Yeah, go on.
Just a quick question about the gradient-based optimization when you're just trying to get the axial polarity.
Yeah.
Is each cell just trying to match, like the cost function is just, if I'm cell 12, I need to make sure my axial polarity is the 12th number in that vector?
Yes.
So when you're doing the back propagation of that loss, that derivative basically only depends on that cell and I guess its neighbors? You're just measuring that instantaneous cost. So you don't need to really get the gradients with respect to all the other cells on the other side of the...
Well, this is back propagation through time. So we take, yes, it is through time. So we start with this ODE model, and then we run this ODE model through a number of steps, and then it forms some gradient pattern. We just back propagate that final state back all the way through the parameters. So it's back propagation through time.
I see. So that's why you are able to get cells on the other side of the chain affecting ones.
Exactly. Yeah.
The other side. Okay.
Yeah.
And the target is that you have a clear anterior and posterior. You don't say that just one of them. Like you're not allow for both. Like you're breaking the symmetry by how you define the target function. Right? You're defining one end to always be the anterior.
Yeah. The target function was, we had a clear anterior and posterior ends, but also in the model, you see here, there is a sort of implicit planar cell polarity, right? So this part of the GRN affects this gap junction differentially compared to how this affects here. So there is an implicit planar cell polarity that's assumed, but I think optimization exploited this to sort of scale it up to the entire network.
And this is biologically motivated in that?
Well, some of the constraints in the model are biologically motivated. For example, this gap junction and the dynamic. Yeah.
I mean the breaking of the symmetry, like the anterior and posterior.
Planar cell polarity is an actual biological feature, but of course it's implemented in different ways. We are just sort of take the inspiration and kind of conceptually use that idea, but not exactly, not precisely, as how biology implements. Go ahead.
Yeah, just I'm trying to understand. So, like you've got these ODs for the Gs,
yeah.
And so you're trying to be faithful to the biology there in terms of how you design those ODs?
No.
That seemed to be what you were saying, that you've got a 7 or how big is that OD?
There is like 9 genes in this network.
I see.
Yeah.
But does it have to be that? Like, what's the minimum complexity you really need over there because it's kind of, I get the kind of hybrid, like, I don't want to be totally phenomenological, I don't want to be totally doing too much detail. How did you kind of converge on, like, why is that the sweet spot somehow?
Yeah, it's in many ways it's arbitrary. In the next example, I will be talking about how, again, that we had a different model for targeting a different problem. There, again, we had some GRNs in that model, but the number of genes we chose there was based on some literature. Again, that number was just the number of genes that we knew about, right? But there are so many genes that we may not know about. This is a number that I just arrived at using just trial and error. I started with 3. Didn't work, and then maybe I scaled it up to 6. With 9, it worked. And so for me, the more interesting thing was the phenomenon itself, the qualitative nature of the phenomenon, rather than the loyalty to biology. But again, some of these constraints are, for example, planar cell polarity and stuff like that was biologically inspired. But that's why it's kind of like a tricky modeling approach. It doesn't make a lot of people happy, but that's what we did. Yeah.
So each of these cells is functionally the same, such that they have to have the same network.
Yes.
But they're not the same left to right. Is that?
Yes, correct.
Okay.
Yeah.
All right. So then that is describing exactly the same behavior.
Yeah, exactly. Yeah. So we only optimize the parameters within a single cell, and that was applied to the entire network. Yeah. Okay. So let me move on to the second example. So this is the phenomenon of bioelectric control of morphogenesis. So in frog embryos, those embryos go through this process called neurulation. So at a very early stage of embryo, there is a 2-dimensional sheet of cells known as the neural plate that then folds into a neural tube. And this neural tube then extends into the spinal cord and then later differentiates into the brain and so on and so forth. So what Weibo Pai in our lab and others discovered in 2018 was this: at this early stage of the embryo, when it has a neural tube, there is a characteristic bioelectric pattern. So what do we mean by bioelectric pattern? So it's basically a spatial profile of cellular membrane potential. So every cell has a membrane potential, and it ranges between about -5 millivolts, which we call as depolarized, to about -55, -60 millivolts, which we call hyperpolarized. So it turns out that the neural plate has to have this V-shaped pattern, bioelectric pattern. So the flanking regions of the tissue have to be depolarized, which is close to -5 millivolts, and the middle portion of the tissue should be hyperpolarized. So there has to be this V-shaped pattern in order for the embryo to develop a normal brain. Turns out if you mess up the pattern, say if you flatten out the entire pattern to being completely depolarized or being completely hyperpolarized, then you get these developmental defects. So we wanted to we want to model this problem. And so what we did was, again, the spirit of minimal modeling. We designed a simple, again, 2-scale model. On the one scale, we have this bioelectric network, and on the other scale, we have this genetic network. So every cell in this model this shows it in a bit more detail. Every cell in the model has a generic pair of ion channels, a depolarizing channel and a hyperpolarizing channel. The depolarizing channel tends to make the cell more depolarized, tends to push the voltages closer to 0. And the hyperpolarizing channel pushes the membrane potential to lower values. And in reality, of course, there are many different kinds of ion channels, but in the spirit of minimal modeling, we just assume these 2 generic ion channels. And every cell, again, has a genetic regulatory network, as in the first model. And we assumed certain arbitrary interactions between how the bioelectric circuitry and the GRN interplay. And we know that there are these bioelectric transduction mechanisms that transduces information about membrane potential into genetic activity, and there is some literature, but again, there are lots of unknowns, so we just assumed certain interactions, and the structure of those interactions were optimized by genetic algorithms, and the parameters were optimized using a gradient descent as before. So we have this two-dimensional model, and we optimize this model to behave in the following way. So if we showed this pattern, where the left two columns are hyperpolar, or it should actually be the other way around. Anyway, the left two columns are hyperpolarized, the middle two columns are depolarized, and the right two columns are, again, hyperpolarized. If we initiate this model with this bioelectric pattern, we wanted all the genes in all the cells to activate. So that's what we optimized the model to do. And if we showed any other patterns, like if all the cells are depolarized, or if all the cells are hyperpolarized, then we wanted all the genes in all the cells to deactivate. So if you think about it, it's kind of a nonlinear problem. It's kind of like XOR-like problem, if you know what I'm talking about. But again, we used gradient descent using PyTorch, and we were able to find a model that does this, and this is how it behaves. So the x-axis is time, and the y-axis is gene expression, and the lines indicate the gene activity for 3 different kinds of bioelectric patterns. If it was endogenous, which is the blue, white, or the red, blue, red, then the gene expression turned up over time, but for the other 2 patterns, they turned down. So the model was able to solve this problem. And again, as before, the question was, how does this model work? In the paper, we actually go into a lot more details about how we made predictions about what would happen if we showed different patterns, and we even verified those predictions in the lab. But for today's talk, I want to focus on how this model works for this particular problem of what happens when we show the model, the endogenous bioelectric pattern. How does it translate a spatially heterogeneous pattern into a consensus of gene activity? Because this spatially heterogeneous pattern drives the gene activity in all the cells to turn up. How does that happen? So to do that, we considered again, a measure of causal influence, but now we considered a second-order causal influence. So this second-order partial derivative keeps track of changes in the gene activity due to changes in pairs of membrane potentials, in the membrane potentials of pairs of cells. Actually, we also computed the first-order causal influence as before, but turned out that it was not statistically informative, because when we compared it with randomized null models, it was about the same. But the second-order causal influence is what stood out from a random null population, and so that's what I will be talking about. So this is a second-order causal influence that keeps track of how genes in some cell in some part of the tissue changes due to changes in the voltages of pairs of cells in possibly another part of the tissue and in a very different point of time in the past. And again, we can compute this and visualize this as networks and see how that network changes over time. And again, for simplicity, we visualize this network only for the strongest causal influences. And this is how it looks like. So as before, at a very short time scale, first of all, I should say that the pattern that we talked about, that I showed you before, has a 4-fold symmetry. So it's symmetrical about this axis, and it's symmetrical about this axis as well. So for the sake of simplicity, you only consider the target genes in the top left quadrant of the tissue, because it would be the same for this, this, and this. So at a short time scale, what happens is that the genes in this quadrant are influenced by pairs of cells that are also pretty much in this area. As you can imagine, at short time scales, the influence do not spread that far. But over longer time scales, a new pattern emerges. So at this time scale, you can see sources of cells that are further away from this quadrant that are influencing this. And it's only this, but not this part of the tissue. Now, at a slightly longer time scale, this influence shifts from here to over here. And it kind of stays negative. But after a while, the influence shifts again back to this area, but now it is positive, and it stays positive for a while, and it kind of shifts back to this area. So the picture that we are getting from this is that we have this locally connected two-dimensional network, but it uses a certain pattern of causal influence that kind of looks like a pendulum. It sweeps across the entire space and changes its the sign of interactions. It switches from being positive to negative. And we still don't know a lot about how to interpret this mechanism, but there are some things that we can tell. First of all, we have this heterogeneous pattern, right? It was like blue, blue, and red, red, and blue, blue. So there has to be some mechanism of integrating all this information across the space. And we think that this sort of causal mechanism does exactly that. It helps integrate information from here to here and back here, right? And it's second order, not first order. And we think that that is because it's using information about differences in the pairs of voltages, not the pairs of the voltages of single cells, to integrate the information and then make a decision. So it's using the second order information and across the space over time. It just sweeps in a pendulum-like fashion, and then it makes a final decision when all the genes are either activated or they're all repressed. So again, this sort of causal network language helps us understand to some extent about how this model solves the problem of spatiotemporal information integration. So I'm going to move on to the next example, but do you have any questions? Yeah.
On your equations a moment ago, what was big V? Sorry, this one?
Yeah.
What is big V?
Sorry?
What's the large V, the capital V?
Oh, that's the voltage of, okay, so V sub j is the Vmin or voltage of cell j, and this is the voltage of cell I, and G is gene activity of gene K in cell L. So there are many different subscripts here, 4 different subscripts. Yeah, it's a second order partial. And it's a partial because we evaluate all this conditioned on the initial state of the network. And that's actually very important because I'll get to it at the very end. And that's actually one of the downsides of this method, because whatever patterns that we infer only applies for a single simulation. We cannot make general statements. The model is the same. The underlying model is the same. But this network changes according to the initial state and exactly how it solves the problem. So that's one of the limitations, but still it gives some insights, I think. Yeah.
So okay, I mean, so the mathematical structure over here is that you've got the system of ordinary differential equations where you've kind of put details of modelings into the G, you've kind of built in the asymmetry of the cells, and you're kind of picking different initial conditions and seeing like what's the sensitivity of the solution at time tau to kind of what's happening, or time t plus tau, depending on what's happening over here. And in a sense, like this interpretation or this modeling was based on kind of the experiments in the lab, if I'm not mistaken, on the slide that you had, like 3 or 4. So I'm just trying to understand kind of how this kind of mathematics is actually capturing the kind of phenomena that you start with. It may just boil down to you saying the same thing again that you said, but I'm not quite sure I can put the pieces together.
Well, so we started with this experimental, right? So this is the experimental result where we found that you have to have this endogenous pattern, right? This V-shaped pattern to get correct brain development. If you change this pattern, you get altered brain developments, and these are defective brains that you get. And so we modeled this by saying that if you input the model with this pattern, so that's the endogenous pattern, then you want all the genes to activate. In reality, it may not be the case that all genes should activate in all the cells. It may be that certain genes that code for brain structures have to activate, but this is, again, a minimal approach. So we just turned this thing into a nonlinear problem, saying that for this pattern, we want on, but for these two patterns, we want off, right? So this is kind of what we started out with, and we designed this model. We optimized this model that solves this problem so that we have a model that we can say, hey, yeah, it sort of represents the qualitative phenomenon that we observed here, right? Starting from here, you get a normal brain. So what we say here is that if you start from here and if you get all the genes to activate, then we think we use it to represent a normal brain outcome. So that's the model we have. Then the question was, how does it, so clearly the model works, right? But what is not clear is how does it work, right? How is it that the model, which is locally connected, able to integrate all of these different pieces of information into a collective gene expression, consensus, right? So there are genes here that know that about this entire pattern. There are genes here that also know about this entire pattern, and that's why they all activate when you see this pattern. So it's a problem of collective decision making, and the question is, how exactly does it do it? And so to understand that, we did this analysis. We made these causal networks. And then I just schematized it and showed that there is some sort of second-order causal influence that sort of spans across the entire tissue. And perhaps by doing this, it's able to collect information across the entire tissue and help make collective decisions in terms of the gene activities. So that's the conclusion here. Again, I know it's detached from biology, and we cannot really use this to say anything specific about the specific model system we have, but we are just using this to say something qualitative about how is it that, at least in this very concrete minimal model, the collective decisions are being made. Yeah.
So you found these models by doing gradient descent?
Yeah.
Is it the case that if you run the experiment again, you get the same model, or are there many models which will work with the same?
Yeah, so that's a good question. And I think I did some of that. And the thing is, to do this kind of analysis is very expensive. In terms of the overall behavior, yeah, we do see something similar from different models. But do they have different underlying causal strategies? It's possible, but it's very expensive to do it. So I kind of didn't really dig into it. So I don't have an answer to that question.
But it would be nice.
Yes. Yeah. So, but yeah. So even for this very simple, like 24-cell model, doing this sort of second-order analysis over multiple time scales was already like taking weeks. So that's one of the limitations of this. So yeah. Yeah. Okay. So here's the last example I have. So this is about electric field guided morphogenesis. And to motivate this, let me show you this video of coming out of some work that happened in our lab almost like 13 years ago. I was not even there in the lab. But what we are going to look at is basically, again, a frog embryo developing over time. But what you're going to see here is bioelectric pre-patterns of the phase of the frog embryo, even before the phase actually develops in morphogenetic terms. So even before the proteins of the facial structures express, you would see a bioelectric pre-pattern. So let me play this. Did you see that face in this? Right, so focus on this embryo. So each one of these are two different, each one of these is an embryo. So you see that there is like a nose-like structure that splits off into a nose and a mouth, and then there is a facial, there's like a ring of fire on the periphery of the face, and then you get the eyes. I don't want you to see these. So these are pre-patterns. I want to emphasize this because these are not morphological patterns themselves. And so So the question is, so this was done like years ago, and but we still don't have a great model that explains how these patterns develop. And so one of the ideas that we had, and so this is work in progress. We are writing it up, and we should have a preprint very soon. But one of the ideas that we had here was that perhaps there is a sort of electric field. So one thing that you can observe here is that there are patterns that form around the periphery of the face, but it seems to trigger things like eye patterns without setting off anything in between, right? So it's not like what you might see in a cellular automata, where signals propagate through space. It somehow feels like there are signals happening at a distance, and they trigger patterns at a different region of the tissue without setting off anything in between. So that got us thinking about whether there may be some sort of electric field-like effect that may be at work. So that's kind of, that's the idea here, where you have this facial features, but you have this outline of an electric field that kind of segregates the different features of the face into their respective compartments. And so the actual pattern is this field plus particles. By particles, I mean the features of the face. So that's the motivating idea here. We designed, again, a minimal model consisting of the bioelectric scale and an electric field. So every blue circle here represents a cell consisting of the same kind of bioelectric circuitry that I mentioned before. And the green circles represent the so-called field grid points, where an electric field force vector is assigned, and every cell will read the force vectors around it to modulate its ion channels. And there is some evidence in the literature saying that electric fields can modulate the ion channels of the cells, and by doing that, it can, of course, alter the membrane potential of the cells. And this is the model in a little bit more detail. So here is a cell, the two ion channels, and it has a set of field grid points, and each point has a force vector assigned to it. And the sum total of the magnitudes of these force vectors determine whether one of the ion channels should open up or should it close. And that's how the electric field modulates the voltage of a cell. And these are just some examples of some visualizations of force field vectors. Here is the simplest example. You have a network of cells, and these are all, the voltages of all of the cells is just 0 millivolts, and the voltage of this cell is negative 55 millivolts. And just by virtue of being negative, it has all the field vectors pointing towards it. So that's the convention in the field of electrostatics. And we have this parameter where each cell can exert influence, field influence through a different range. So it doesn't have to be the case that it exerts the field influence throughout the tissue. The influence can be contained within a small square. So that's what the red squares indicate. And here is a case where you have different patterns of voltages, and depending on the range of the field, you get different field vectors. So one thing to notice is that if you take a cell somewhere like here, the cells around it have about the same voltage as that cell. So just by virtue of that, the force field vectors around it are about 0. So you only get force field vectors when there is a sort of gradient in the pattern. So that's something I just wanted to show you about. But again, this is a detail that may not be relevant. In any case, so what we did with this model was the following. Initially, we thought of training or optimizing this model to develop a phase pattern starting from uniform conditions, like what we did in the first example. But the phase pattern is a bit complex, right? It has different features. It's a nonlinear pattern. It's not like a simple linear gradient pattern. So having the model develop a complex pattern like this from uniform initial conditions turned out to be tough. So what we ended up doing was, again, in the spirit of minimality, we designed a paradigm where we just stimulated the field around the boundary of the tissue, and we used optimization to infer the oscillation parameters of the stimulation. And we just do that transiently. So we just do it we stimulate it for just 10% of the time, and then let the network go, and the network will just bootstrap the phase pattern after that, following the transient stimulation. And we were able to infer parameters to get the network to do exactly that. So here's an example of how the network does that. So initially at time step 0, we start simulating only the boundary. Again, so this is like controlling only the boundary, and we don't go into the bulk of the tissue at all. And at the end of the stimulation, we get this pattern that doesn't look anything like the face at all. But if you let it go after this, slowly what happens is that you get this ring of fire that we kind of saw in the first video, and that sort of triggers features or cells in the bulk of the pattern, of the bulk of the tissue, slowly building up the facial structure. Again, this facial structure is not as perfect as what we saw in the video at all, but you get a good enough example, I think. Starting from this to get here, I think it's very interesting. And it kind of suggests that there is a sort of shepherding-like strategy where the boundary sort of shepherds the cells in the bulk into forming the phase pattern. And so in that sense, it is I think it's a collective a behavior problem. And here is a video of how that happens. So initially, you see these things oscillating because it's being stimulated. But after a certain point, it will just stop. The oscillations will stop, like here, and then slowly the pattern will develop over time. So you don't see any features in the bulk. But let me just speed it up. Over time, you see features forming gradually. And you see this sort of nose, a thick nose that forms initially, and then it splits into a nose and a mouth, very similar to what we saw in the video. And then it converges to almost like a face-like pattern, right? My wife, she thought when I was making these videos at home, she thought I was playing video games. So she actually traveled all the way from India to make sure that I'm working and not playing video games. So she's right here. So anyway, I did ran some causal network analysis as before, but the first order analysis. And again, we saw something interesting. So this is almost the last slide here. So at a very short time scale, so these are longer time scales. At a very short time scale, the causal influence is locally constrained. But as time goes, you see these beautiful patterns emerge where one unique feature of this causal network is that the cells on the boundary is what seems to be driving the activity of the cells in the bulk, right? You see almost all the source cells lie on the boundary. We are still sort of unpacking this, but one thing we found was what this predicts is that the shepherding strategy that we are talking about is the boundary that's shepherding the cells in the bulk. And so that predicts that if you alter the states of the cells in the boundary, the pattern should mess up. And that's what we actually find. And we also ran some information theoretic status measures between the bulk and the boundary and things like that.
What would that mean in the biological setting? Yeah. Like, I see the boundary.
Yeah, so the biological, if you go back to that video I was showing you, what it would mean is that, again, it's somehow, like, let me go to this point where you see this ring of fire that I was talking about here, right? There is this ring of fire right here. that may be the ones that's triggering activity, that may be the ones responsible for the formation of ice. And it's not like a local activity is somehow triggered by the ring of fire on the periphery. So that's a prediction that holds out for the model, but we don't know if that's what's happening in biology. I guess from a high-level perspective, what it means is that a lot of patterning happens in the long range. Like you might think that if you see an eye forming, it may be due to the gene activity in that region, but it may be the case that it's actually being triggered by cells far away. So again, in the language of collective causal networks, we see a model that started from this, and we get these sort of patterns that we can now use to interpret how it works, how this model facilitates patterning or formation of this vertebrate phase patterns, right? So basically, in summary, I showed you these 3 examples of using this framework of causal network to infer this sort of high-level causal patterns that we can use to think about how collective behavior works in disparate systems, at least in this case, disparate phenomena in developmental biology. So, but of course, there are downsides to it. As I mentioned before, these causal networks that we infer only works for the very specific simulation that we ran the metrics on. And so we cannot make any general statements, and making predictions also becomes difficult. Because if we make some predictions, then those predictions would have to be at the microscale, at the level of the individual cells. So if it means that you have to change the cells to a different state, then it may lead to a different macroscale pattern. So that's one of the downsides. But again, I think, as you saw, as I hope I convinced you guys, there is still some insights that you can draw using this kind of framework and using this kind of high-level common language. And also, it's very expensive to compute because we use these automatic differentiation methods that makes use of a lot of space and time to compute this. So there are some downsides to it, and maybe in the future, we can try to improve this better. So that's the end of my talk, and I would like to thank Mike, my boss, who inspired me with a lot of ideas and also paying for my work, and Pi, who I collaborated with for the second model that I showed you. But that's it. Thank you.